Assalamu alaikum warahmatullah. I'm Alzahraini, respiratory specialist intern. I'll be the presenter of today's topic, which is about neonatal lung development. Our general objective is to describe the 5 stages of lung development over a timeline. I will start by presenting the basic physiology so that you can comprehend what will be coming soon. The embryo is made up of 3 distinct germinal tissue layers that ultimately form all tissues and organ, and therefore organogenesis established from 3 types of cell division. We have endoderm, the innermost layer of a cell that forms the canal of any organ, such as the epithelium lining of the trachea and bronchi. The mesoderm, the middle layer of a cell that forms the tissue-like organ, such as the visceral pleura, the lung tissue, and connective tissue. And ectoderm, that forms the nail, hair, skin, etc. Therefore, we expect the lung tissue to be formed by mesoderm. Thus, any anomalies in the lung tissue is related to mesoderm malformation or deficiency. Another basic physiology that I want to manifest is the lower respiratory system, which divided into conductive zone responsible for gas conduction, start from the trachea, right and left mean stem bronchi, secondary bronchi, aka looper, tertiary or segmental bronchi, terminal bronchioles. Then respiratory zone, responsible for gas exchange, composed of respiratory bronchioles, alveolar duct, alveolar sac, and alveoli. The lung develop in the same sequence that I mentioned, thus you should expect the alveoli to be the last to develop. Progression of lung development. The lung process through initiation, branching morphogenesis, and differentiation. In prenatal life, lung develop over 5 stages. The initiation of the lung start with the first stage, which is known as embryonic stage, at gestation age of 3 to 6 weeks during embryonic period. Lung first derived forming a bud from the ventral foregut endoderm at the caudal end of the primordium. It enlarges, bifurcate, giving rise to left and right bronchial buds, which is known as mean stem bronchi. During the 5th week of gestation, a second round of branching results in 3 secondary buds in the right lung and 2 in the left, becoming loop in a mature lung, as you can see in picture 2. Ultimately, a 3rd round of branching gives rise to bronchial tubules that will become the bronchopulmonary segment in a mature lung. At the same time to this event, the cranial portion of the primordium gives rise to trachea and esophagus by the end of this stage. Since initially, the lung bud is in open communication with the foregut, as you can see in picture 1b, the tracheoesophageal ridges separated from the foregut, fusing to form a septum, divided into dorsal portion, esophagus, and ventral portion, the trachea. This takes me to one of the deformities that occur at the embryonic stage, which is tracheoesophageal fistula, which is incomplete separation of the esophagus and the trachea by tracheoesophageal septum. Since we mentioned deformities, another embryonic stage malformation is underdeveloped lung. That might come in form of agenesis, absence of the bronchus and the lung, which might occur bilaterally or unilaterally. A unilateral agenesis is compatible with life. The remaining side compensates by hyperexpansion. Also, it comes in form of hypoplasia, which is reduction in number of lung cells, airways, alveoli that result in a lower organ size and weight. The reason why I mentioned some example of malformation is to illustrate that birth not necessarily means a well-developed lung. This is the tracheoesophageal fistula. It comes in 5 forms, such as in image A, there is fistula with the distal part of the esophagus. And this is the bronchial hypoplasia. The second stage of lung development is pseudoglandular stage. Now the embryo is a fetus at gestation age of 6 to 16 weeks. The lung have a gland-like appearance, and the previous formed bronchial tree branches. Also, there is branching of the pulmonary vasculature, and type 2 pneumocyte appear. Therefore, we expect the surfactant to be secreted in the third stage. There will be also development of the cilia, mucus gland, and goblet cells, formation of terminal and respiratory bronchioles, and by the end of pseudoglandular stage, the conductive zone end. The Canalicular Stage is the third stage at gestation age of 16 to 26 weeks. There will be formation of lung periphery, respiratory bronchioles develop from the terminal bronchioles, increased vascularization, cause invasion of capillaries into the mesenchyme surrounding the acini, and leading to air-blood interface formation. Type 2 pneumocyte differentiate to type 1 pneumocyte. Lamellar body formed in type 2 pneumocyte, thus beginning of surfactant synthesis and secretion. First breathing movement can be registered, and fetus are able to survive outside the uterus after roughly 22-24 weeks of pregnancy because surfactant began to synthesize. Saccular stage at gestation age of 24-36 weeks. Lengthening and widening of saccule will occur. Blood-air barrier became well developed, and surfactant detected in the amniotic fluid. Thus, surfactant production is sufficient enough to prevent atelectasis. And this is why premature infants are prone to respiratory distress syndrome due to insufficient surfactant, since the sufficient surfactant present after 35 weeks of gestational age. Alveolar stage, the last stage in fetal life at gestation age of 36, that continue till 8 years. There will be formation of immature alveoli, and 50 million immature alveoli formed at this stage. Alveolarization of the lung after birth occur with a secondary septal formation from the primary septum formed in the prenatal life. In picture A, the lung parenchyma made of saccules, and in picture B, saccules transformed into alveolar duct and alveolar sac. Term infants have approximately 50 million alveoli, which undergo increase in surface area and number, increasing to 300 to 400 million at maturity. After birth, the alveoli develop increasing in number until the age of 8, and increasing in size until growth of chest wall is finished. And that's it for today's topic.